for being here. Yeah, so the title of my talk today, Exploring Drivers of Spatial Non-Stationarity and Wildlife Relationships through Species Distribution Modeling. So I have a lot to talk about, and I'll try to keep it within the time limits that we have today. Let me get my slides advancing. Okay, just as a primer for the talk today, we're looking here at a range map of American black bear across across North America. This species exists just outside, or in the Santa Monica Mountains of California, just outside of LA and 10 lanes of traffic. Of course, north to the tundra environment of the Yukon, and along the eastern seaboard of eastern United States. Specifically, I'm thinking about the Pocosin shrubbery of coastal North Carolina, into the Appalachian Mountains, in and out of Asheville, and even down just outside of Orlando, Florida. So when we ask, pretty basic, typical questions in ecology, such as what is the effect of housing density on black bear abundance and distribution? Or what is the effect of forest cover on black bear abundance and distribution? Here, of course, is a black bear in eastern North Carolina and the agricultural landscapes out there. Hopefully you're thinking to yourself at this point, given these few slides, that, well, maybe it just depends. It depends what the effect of housing density is. It depends what the effect of forest cover is. And I wonder, why? Why does it depend? Can we just credit, this is just an omnivore generalist species and it can do whatever it wants? Or maybe there's some sort of genetic differences across space that might be driving differential use of housing or housing densities or forest cover. Maybe it's some learned behaviors that they're locally adapting to for an environment in which they are. Of course, it could be strong human influence driving these differential uses across space. And maybe it's just all resulting in some change in the species' ecological niche. Well, again, just to remind you all, even though it's a foundational concept in ecology, I just want to remind you of the ecological niche. And here I'm specifically talking about what's called the fundamental niche. There's two flavors. Here's the fundamental niche, and that just describes the requirements of a species to exist in some given environment, right? So for example, if we have some gradient of temperature and precipitation, taking on all values of temperature and precipitation possible, maybe some species just exist in some subset of the possible conditions of those climatic gradients. The other flavor, in contrast, would be what's called the realized niche. And this takes into account both those abiotic, the realized niche is a function of both the abiotic or the environmental conditions, as well as biological interactions. So predation, competition, mutualisms. So the realized niche describes the conditions where the species actually occurs within its fundamental niche. And typically, but not always, that realized niche is a subset or smaller than the conditions of the fundamental niche. So following up on the slides I just showed, I kind of want to think about, are realized niches constant across space? Are animals using the environment the same across large space? We see animals, or even some given individuals of a given species existing in very different environmental conditions across geographic space, but it's unclear what's driving this variation in the realized niche. In other words, are these niches that we estimate in some given space sufficiently predictable to be readily transferred from one location to another? So within this, I'm going to explore this with a couple case studies in North Carolina and beyond. But before I do that, I want to introduce two more concepts of the niche. So I want to create some contrast in what's called niche conservatism versus local adaptation. And this is related to how niches may or may not change over space or time. So niche conservatism would describe the tendency for a species to retain those traits of their ecological niche through space or time. And that would look something like this. So here again, we have this niche and these environmental gradients. And on the far left, we're showing 60,000 years ago. In the middle, it's 10,000 years ago. And on the far right, it's present day. So we're saying that the niche conservatism would say that this species niche is remaining constant through time. Of course, local adaptation would be a scenario where there's some deviations from the expected niche due to surrounding ecological context, anthropogenic effects, or potentially both. And what that would look like is that we have some shift or change in the species niche through time or space. So that's what we're going to pursue today. And again, we're going to have a couple of case studies focusing on what I'm calling these spatial dynamics and species-environment relationships. I'll first start with a case study on white-tailed deer in North Carolina. I'll then move to a continental-wide model of American black bear. And finally, I'll very briefly describe where all of this is going in terms of combining multiple data sources and species distribution models. And for that, I'll briefly introduce a scenario of using, or example using North Carolina's mesocarnivores. So getting right into it, again, we might need another primer just so that we're all on the same page. I'm going to be talking a lot about species distribution models, so I want to make sure that we're all on the same page again. So the fundamental purpose of a species distribution model is to relate observations of animal or plant occurrence to environmental conditions through geographic space. And when we do that, we specifically quantify these, what I'm calling species-environment relationships. So we can quantify the effect that forest cover or housing density is having on American black bear. Is it some positive or negative effect? And from these estimated relationships, we of course can predict just the distribution of the species given environmental conditions, or even changes in the species distribution under different environmental conditions, climate scenarios, or land changes in land cover. So why should we model species distributions? Well, of course, the simplest reason is just to understand these relationships between a species and its abiotic and biotic environment. This has been the focus of some of ecology for at least a century now. But SDMs, or species distribution models, have many other applications as well. They're widely used in conservation planning to prioritize or target areas for protected status. They can also be used to predict the impact of some natural resource management activity or some proposed land use change. They're also widely used as a preventative tool to predict where invasive species are likely to establish. And of course, finally, human-induced environmental change has many implications for species distributions. And SDMs are just a tool to predict how species or how distributions might change under a range of climate scenarios. So if we ask, again, coming back to a basic question, is what is the effect of forest cover on deer distribution in North Carolina? We could use a species distribution model to estimate this effect. So we have some occurrence records of the species, the presence or absence of deer across the state. We have information on forest cover across the state. We can estimate these effects of forest cover on deer distribution. From another view, say we have records of this, we have some field records or the animal's presence or absence. And we also have maps of the environment, say forest cover, temperature, or maybe some elevational gradient. We put those through a statistical model, and then we get the output is some probability of species presence. Let's see here. If we focus on the statistical model, typically regression is the underlying workhorse of the SDM. And that's here, just to remind you all what that looks like, we have some regression line down there, where we have the response Y, the presence or absence of some species. We have the covariate here, forest cover, and we want to estimate the effect. So we have two parameters here. We have an intercept term represented by B sub 0, and the effect of forest cover represented by B sub 1. Typically, this is done with using logistic regression. So we estimate these terms either using maximum likelihood or some Bayesian inference. And we find that the effect of forest cover is 0.5. It's a positive effect of forest cover on the species occurrence. Well, what regression assumes is what's called stationarity. And that just means that these parameters that we just estimated are consistent or constant through the region of interest. That means that if we're focused on the triangle, that the effect of forest cover is positive and constant on white-tailed deer across this entire range. If we have animal observations across North Carolina, that means that the effective forest cover is positive and constant on white-tailed deer across all of North Carolina. And you can probably see where this is going. If we have observations on deer occurrence across all of the United States, we would assume that that effect of forest cover is constant and a 0.05 across all of North America. And that's probably just not realistic for a number of reasons, but namely, think about all the different environmental conditions that animal is exposed to across this entire range. Think about the different human pressures that the animal is exposed to, or even the biological interactions that some species is exposed to. So it seems likely that the animal is going to be using the environment differently across space, and that effective forest cover is probably not going to be that consistent, constant 0.05 if we were to estimate it in different areas across this range. So rather, what I'm suggesting or describing here can be termed as spatial non-stationarity. And that just describes, and that just means that there's variation in the direction. So is it a positive or negative effect, and or magnitude of the effect across space. So we see some differential effect of, for example, forest cover across space. So revisiting this question again, we have our animal occurrence records, we have forest cover. So if we want to know the effect of forest cover, typically what is assumed that it's a stationary effect that's constant across all of North Carolina. It's the same effect of forest cover. What I'm proposing, however, is that there's some differential effect. And I think it could come in a couple of different flavors. Here I'm saying that I'm suggesting that maybe it's some coarse scale variation in the response of forest cover. So here I'm showing the three primary physiographic regions across North Carolina. It's the mountains and the Piedmont, and the coastal plains. So that would suggest that the effect of forest cover, or animals are using forest cover differently across these three primary regions. Similarly, it could be some fine scale variation in the effect, and something that's perhaps operating more at some population level, or just something that's not easily delineated by these well-known boundaries, such as ecoregions. And non-stationarity in ecology is probably very common, but it's primarily largely understudied, and I think it's understudied for at least two reasons. One, it just definitely requires these large-scale, dense observation networks of animal or plant occurrence. And then secondly, it's typically pretty computationally and or statistically demanding. It's not readily available. There's no really button-pushing approaches to estimating these effects. So given that it's largely understudied, there are many outstanding questions in ecological non-stationarity. One, primarily just the types of species. For example, are they specialists that have non-stationarity? Are they generalists? What type of ecological predictors? We're thinking about climatic responses or land cover conditions. The resolution of the non-stationary patterns, so as I just showed here, that fine versus coarse scale. And importantly, if non-stationarity does exist, what is driving this variation across space? Well, I can think of at least a few reasons where there might be non-stationarity or drivers of non-stationarity. One, we could think about just simply resource availability. So here we're looking at, on the left, landscape, primarily agricultural landscapes with remnant forest patches, and on the right is contiguous forest cover. So if forest cover is needed for this species during their daily or monthly or even annual needs, we would expect that species on the left-hand side would be more drawn or more strongly attracted to those remnant forest patches in the agricultural landscape. In contrast, if the species is moving through this large contiguous block of forest cover, they might be sorting out different types of land cover types, agricultural, for example. So then in that case, we might actually see a neutral, potentially even a negative effect of forest cover if we estimated some relationships in that zone or in those conditions. I also think differences in biological communities can drive differential use of land cover, or excuse me, of landscapes. Here I'm illustrating the idea of mammalian predators driving differential use of deer. And that is that areas where predators are present, or even we could look at abundance gradients, we might see differential use of deer by the landscape based off of some sort of predation risk that they have in these areas. Finally, of course, it could be something just about the species biology or the species populations across some region of interest. Here I'm representing this by the 5 primary deer management units in North Carolina. And across these management units, we have differences in deer size, male-female ratios, even the timing of gestation periods. So there's just something about the deer in these areas that also might lead to differential use of the land cover across space. Of course, all of these could be interacting to be drivers of non-stationarity. So for this specific case study, I first and foremost, of course, wanted to estimate the distribution of the focal species across North Carolina. I wanted to test for these patterns of non-stationarity at the state level, determine the types of ecological predictors exhibiting non-stationarity, and then finally, I wanted to explore potential drivers of these non-stationarity patterns. And for sake of time, I won't be able to get to this last bullet point, but we could chat about it afterwards if folks are interested. So I mentioned that we needed some dense, large-scale observation network to get at this question. During 2016 to 2019, there was a effort led by Dr. Roland Kays and the North Carolina Wildlife Resources Commission called the North Carolina Canid Critters Project, where volunteers distributed cameras across the state under a standardized sampling protocol. And this resulted in an unprecedented effort. It was one of the largest scale camera trapping efforts to date. Over 4,000 cameras were deployed in North Carolina, resulting in about 70,000 days of search effort. Okay, again, just to remind you all, we're focused here on deer and we're interested in the occurrence probability. That's related to the distribution of the species. So what is the probability that some species or this species is present in a given area? And you might be thinking to yourself, well, deer are everywhere, and that is true. But I've divided North Carolina into 5,500 grid cells that are in about an area of 25 square kilometers. So we're evaluating the occurrence probability in each one of these grid cells based on the differential environmental conditions within each grid cell. To think about the type of ecological predictors here, I just focused on some landscape composition metrics, such as the percent forest cover in a grid cell and the average housing density in a grid cell. But I also looked at landscape configuration metrics, such as patch richness density and contagion index. Patch richness density just describes the number of land cover types in a given grid cell, and the contagion index describes the kind of the shape or the size of those land cover patches within the grid cell. To test for these patterns of spatial non-stationarity, I fit 3 models to deer and other species across the state. I fit what I'm calling a standard GLM. So that's just the typical approach that I described earlier, where we're assuming some constant effect. Then I fit here what I'm calling that ecoregion model, and that's getting at that coarse scale non-stationarity patterns. And then finally, I fit what's called a spatially varying coefficients model to estimate these fine scale effects where we don't actually know the a priori delineations of how animals are behaving differently across space. And just to remind you, the graphical here on the far left, we have that constant effect. So the standard GLM, in the middle would be the ecoregion model, and on the far right would be the spatially varying coefficients model. So for example, in the ecoregion model, if we're thinking about the effect of forest cover, we'll get 3 estimates of forest cover, 1 for each primary ecoregion. Same with the housing density, the effect of housing density in the mountains versus the coastal plains, for example. And then in the spatially varying coefficients model, we'll actually get 5,500 estimates of forest cover, housing density, and those 2 landscape composition metrics. So 1 estimate for each grid cell. So just to bring back that equation I highlighted earlier on, it pretty much stays the same for the spatially varying coefficients model, but instead now, each grid cell indexed here by the letter I gets its own estimate of intercept and its own estimate of, for example, forest cover, along with the other metrics I just described on the previous slide. So moving a little quickly to results, again, I mentioned I fit this for several species, but just focusing on deer here, we found that the spatially varying coefficients model, or I found that the spatially varying coefficients model was the best fitting model and it had the highest predicted performance. So I realize there's a lot of numbers here, but just know that this column right here, I evaluated WAIC, which is just a measure of model fit. And this is the change in WAIC. So the SVC is the top model. And then this is how different the other 2 models were compared to the top model. And then I also have the AUC and our area under the, it's just a measure of predicted performance as well as a Breyer score. And I'm just putting these up here to illustrate that even though the SVC model was a top fitting model, it had comparable predicted performance to the other models, which is actually a good sign. Typically, if we, since it had a relatively good predictive performance, we can kind of rule out that there's some overfitting occurring here in this modeling fitting process, which can be quite common in these spatially varying coefficients models. Okay, so the SVC, so deer appear to be behaving differently across space, and it appears to be occurring at a fine scale, or at least not one described by the ecoregion boundaries. And then we might want to think about, again, to see which type of configuration or which type of land cover metrics are deer responding to differently across space. Here I use what's called a stationary index, and basically this value is greater than 1 would indicate non-stationarity. We see that with the landscape configuration metrics, there was actually a relatively low signal or no support for non-stationarity, and it was just the forest cover effect, the effect of forest cover, the deer tended to respond differently across space. So let's dive into that a little closer. Of course, we can get from, of course, in the species distribution model, we get the output of these spatially varying coefficients models are what we're most interested in, the occurrence of the species across space. Here, darker colors suggest a higher probability of occurrence, and lighter colors suggest a lower, or indicate a lower probability of occurrence. So you can note that, I'm not sure you can see it on your screen, but the lowest that would ever get is around 0.2. So yes, deer are indeed everywhere, but based off of the environmental conditions we used, that we do see some variation in the probability of occurrence across space. And I just also want to remind you of the actual forest cover across North Carolina. We see out in the western part of the state near the Smokies, we tend to have a really high forest cover in each of these grid cells, and that decreases as we move towards the coast on the east. Now, the really interesting result of the spatially varying coefficients model is, again, this estimated effect of forest cover across space. So now each of these grid cells are representing that actual regression coefficient that we estimated in the model. And we can see that it ranges from -2 up to +3. The lighter blue colors and gray colors indicating a negative response to forest cover, and the darker colors, or the trend towards red, indicating a positive response to forest cover. So out in the far western part of the state, we see that the probability of occurrence actually decreases as forest cover increases. And here in the Piedmont, the central part of the state, the Piedmont zone, where it happens to be the most populated part of the state, we have a positive effect of forest cover. So the probability of occurrence increases as forest cover increases. And again, I mentioned that we explored this a little bit further in depth in the chapter, and we found that there was some interactive effect of deer biology and predators driving this differential response of forest cover across space. And again, for sake of time, I'm not gonna be able to get into that, but we're finding that we can get this broad scale with these spatially varying coefficients model. We can still get this broad scale inference that ecologists are interested in. We can estimate distribution across large space, but we can get these management level relevant results that can still inform deer management and conservation, or species conservation at a resolution that's able to be managed at. Okay, so moving on to case study 2. In North Carolina, I found some support for ecological context and human pressure as a driver of non-stationarity. However, this was only at the state level, and I was interested in what might be a driver across an entire species range. So again, as I and recently others have found support for ecological context, some have found that genetic differences can be drivers of non-stationarity. So in this chapter, I wanted to kind of focus a little bit more on these drivers and using black bear across the entire North America to focus on this question. So just to remind you all of the difference between niche conservatism and local adaptation, since I'll be focusing on that in this case study. The niche conservatism describes a scenario where an animal is behaving, or it's the tendency for a species to retain their ecological traits through time or space, and that local adaptation would describe deviations from this expected niche, typically due to surrounding ecological context, anthropogenic effects, or both. So if niche conservatism is driving species environmental relationships, then the way the species uses the environment should be strongly predicted by some evolutionary history, perhaps described by some Pleistocene refugia lineages. Here on the left, I'm showing the 4 estimated lineages of black bear across the continent. We can see that there's an eastern lineage that takes up most of the species range, and then there's 3 smaller lineages along the Intermountain West and the West Coast of North America. However, as you can see, an individual lineage, however, can inhabit a wide range of environmental conditions, and there might be some differences within that range, even though they might, differences within the range. And finer scale delineations may be more appropriate. For example, although directly related to the lineages, some varied relationships may be better described by these subspecies boundaries, which are, of course, are nested within these larger lineage subdivision. And they would still suggest that there's genetic differences driving the differences in species-environment relationships. In contrast, if we have local adaptation as a better explanation of species-environment relationships, then maybe, again, the ecological context represented here by differences in ecoregions across space, or maybe it's differences in biological communities that might be driving differential use of the land cover. Here I have shown the range maps of the top or 4 top predators across North America. We've got brown bear, gray wolf, cougar, and then the sad remaining patch of the red wolf out here in eastern North Carolina. So if it's local adaptation, maybe it's something about this that might be explaining the variation. So in this chapter, I kind of use these 4 hypotheses as potential drivers of non-stationarity. And in this, again, some of the primary objectives in this chapter were just to explore the evidence, test for evidence of spatial non-stationarity across the species range, and then look at these 4 hypotheses about drivers of ecological non-stationarity, thinking in contrast of niche conservatism versus local adaptation. If you're thinking back to the first chapter, how I told you that we need dense, large-scale observation that works together this question, you might be wondering, how am I going to pull this off across continental North America? Well, we're no longer in a data shortage in ecology. Here I'm showing just mammal observations during 2015 to 2019 on the iNaturalist database. iNaturalist is an online social network for sharing biodiversity information to help others learn about nature. Observations are submitted to the database by iNaturalist users, and the iNaturalist community identifies the observations to the highest taxonomic resolution possible. So for example, species or subspecies. And then iNaturalist users can submit observations across all taxa anywhere on Earth whenever they want. But that being said, again, just in the past 4 or 5 years, there were about 370,000 mammal observations. And of those 370,000 mammal observations, we had about 6,500 observations of black bear in this database. So I use the iNaturalist database to look at these potential drivers of non-stationarity. For fitting the species distribution model, I focused on 8 ecological covariates across space. Here I've switched over, instead of the NLCD class, I've switched over to the MODIS land cover product, where the MODIS product has 3 different forest cover classes that we could think about. So conifer, deciduous, or mixed forest type. I also estimated effects of cropland. That was derived from the MODIS product. I looked at mean annual temperature and precipitation, and then values of human population density and protected area status. And similarly, as I've done at North Carolina, where I created a mesh of grid cells, I did that across North America, and that resulted in just over 500,000 grid cells, about 502,000 grid cells across all of North America. And I used those at I created those at an area of 50 square kilometers. So I evaluated all of these ecological covariates inside of each grid cell, and I summarized the iNaturalist data within each grid cell for the model fitting process. So to test for patterns test for non-stationarity, I used a very similar approach as case study 1. And just to remind you all, of course, the traditional approach would be use all observations in a single model, and we estimate the effects of those 8 ecological effects across space. And this would result in a single estimate of each of those ecological effects. So for example, there'd be one single estimate of deciduous forest cover on black bear. We would get some point estimate in the measure of uncertainty represented here by standard error. And although I really tried hard to use the spatially varying coefficients model again, to fit it at this resolution, it would be, at least for me, computationally unfeasible. So I used what I call a local spatial window approach, where I create basically little windows across the entire range and fit submodels to each of those windows. So for example, if we just have a window over this, so here's a window here, I would only look at environmental conditions and animal observations inside of that window and estimate effects inside of that, just that window. And I did this across the entire range, and I actually did it with quite a few windows. Here's looking at what 50 windows look like, but I actually used over 1,200 windows. So I fit over 1,200 models to these animal observations across space. You see that there's tons of overlap, and that's actually intentional because eventually these models, it's kind of what's called an ensemble modeling approach. So each of these windows that are overlapping, you eventually average the values that you find, the estimates and the predictions you find in those windows to scale up and make predictions across the entire scale. Again, I use that kind of stationary index to find support of non-stationarity. So this approach, of course, results in many estimated effects of a given ecological covariate. Here, if we're just looking at, for example, the effect of protected area status, this would be the traditional effect where we have some point estimate and some uncertainty measure. And then this is called a violin plot, where we look at the range of effects in each of those 1,200 windows. So we have a large portion, for example, being a positive effect, whereas some areas we might think about a negative effect of protected area status. And I'll get into that point there. But just as a check marker to see whether whether iNaturalist database is giving us information that we think is as accurate and known of the species, I just want to look at estimates from this traditional approach, so the global model estimates. And we're finding estimates that we would expect to see. So for example, overall negative effects of cropland, positive effects of the 3 different forest cover types, a negative effect of human density, very strong relationships with protected area status, positive precipitation effects, and kind of neutral or no effect of temperature. Sorry, I didn't describe this. The regression coefficients on the y-axis, and those 8 ecological covariates, of course, on the x-axis. So the traditional approach, we are finding things that we would expect to find with black bear across their entire range. But this local window approach results in quite a few different effects of forest cover, or excuse me, for example, here just focusing on that protected area status, I showed you the violin plot earlier. What that looks like in space is that we have, and each of these are the centroids of all of those boxes that I showed you. So I'm just representing the centroids for a little bit of clarity in the map. But we see the red colors representing positive effects, the blue colors representing negative effects, and the white colors represent a neutral effect of protected area status. So we see some differential use or differential effects of protected area status across space. For example, if we focus on this blue area, you might be wondering, why would there be a negative effect of protected areas on black bear? Well, here, just to orient you, I've labeled Little Rock, Arkansas. I've labeled Little Rock, but we can see that there's all these orange dots represent the actual observation, and these gray blocks represent the protected areas across space. So even though we are getting observations in the protected areas, there are many, many observations of black bear outside of this protected area. So when fit to just a window around these observations, we find some negative effect of protected areas. And that's just suggesting that animals are using different space outside of the protected areas in this region. In contrast, along the Intermountain West, we see largely positive effects, and we see that most of the animal observations of black bear are occurring indeed in these protected areas along the Intermountain West. And again, I have labeled Wyoming and Montana to orient you a bit. So that's kind of what's going on with these. And if we look at all of those 8 ecological effects, it's really interesting. We could probably spend an hour just talking about this slide about what might be driving differential black bear observations across space. But we can estimate these effects across all 8 ecological covariates. And you can see just based off the same color pattern that there are indeed negative and positive effects across each of these ecological covariates. Thinking back to the ensemble model approach, how I told you we can scale up. Of course, we can get what we're interested in, and we can generate estimates of iNaturalist black bear intensity across space. I have illustrated here in the dotted line the current IUCN range map of black bear, or the International Union for Conservation of Nature. And we can maybe start to think that, well, first we see that the distribution or the estimates of iNaturalists are matching pretty well with what we know about the species distribution, but potentially also illustrating some areas outside of the current range, at least on the IUCN database, where black bear are probably occurring, or definitely are occurring. So it's a really interesting result, and it's an exciting result to show this is kind of one of the first approaches using iNaturalist for this, and it's exciting to see that the database has some important information about the ecology of species. Quickly circling back, I wanted to illustrate again these potential drivers of non-stationarity. So I showed you all the 4 hypotheses I had, and in the center now we have each of these gray dots representing the centroid of those boxes. So for example, if we have a box in the panhandle of Florida, it would be assigned to the eastern lineage, the Floridanus subspecies, no competition, and the eastern temperate forest. So then I would use all 1,200 of those effects of, for example, protected area status, and regress it against each of these hypotheses. So each one of these centroids was assigned to one of the subdivisions of the hypotheses. And what I found, well, first off, I found no support for the Pleistocene refugia lineages as an explanation in the variation in species environmental relationships, nor the competition hypothesis. And largely, I found the most support for the subspecies delineations, specifically focused on the modus products and the land cover products and the anthropogenic effects. And then the ecoregions best explain the variation in response to mean annual temperature and mean annual precipitation. So this would suggest largely there is niche conservatism represented by the subspecies subdivisions driving how animals are using this, or how black bear are using the environment across large space. But we also see some perhaps local adaptation to temperature and precipitation, which is represented by the difference, the ecoregions. And that would agree with the predictions that bear are likely not very vulnerable to forecasted climatic changes. So this is interesting results. And again, there's lots to say here, but unfortunately, I'll have to keep moving on for sake of time. Happy to answer questions if we have it. Okay, so in those 2 case studies, I found pretty overwhelming support for ecological non-stationarity, and I found some interesting patterns in how animals are using the landscape differently. Finally, I want to quickly focus on this idea of combining multiple data sources in species distribution modeling. It's a concept, it's a modeling approach that's been around for about 5 or 6 years now, with a lot of great promise in ecology. And as you're starting to see, or at least get the idea, we're no longer in a data shortage. So we're thinking about how can we make best use of the available data. Focusing back real quick as an example on the black bear, here shown in North Carolina are the Canada critters observations. And then we have other efforts across the United States. Here I'm showing Snapshot USA, which was a coordinated camera trapping effort. We have GBIF, or Global Biodiversity Information Facility, which is another open access database. And again, the iNaturalist database. So we have tons of data available about species species occurrence, but all of these different data sets have their own nuances and potential biases. So we need some modeling framework where we can incorporate information from all available data, but account for the nuances or biases in each individual data source. And that's what's been largely being developed, and it's termed integrated species distribution models. So again, there's large potential to combine what I'm calling here structured and unstructured data. And that just is data that's collected under some specific sampling protocol versus opportunistic presence-only observations such as GBIF or iNaturalist. And there's this increase in readily available data sources. So with ISDMs, we want to be able to recognize and incorporate differences in data collection methods and responses whenever we combine all of these data together in a single model. And we want to be able to take advantage of the structured data that's collected under a robust data collection procedure to use as a benchmark whenever we incorporate data that perhaps has more limitations. And finally, ultimately, we want to improve our inferences by getting a more holistic picture of how the animal is distributed across space by incorporating multiple data streams. So even though this is very promising, we're still in the early stages of ISDM. So there are many, many open questions. And the one I focused on in this section was, can we identify features of the data to know when integration is appropriate? This is typically a very computationally and or statistically demanding procedure again. So it would be good to know whether, before we get into the modeling process, of whether two different data sources should even be integrated. So we might think about ratios or differences in the amount of structured versus unstructured data. We might be able to think about how the correlated data sources might affect our results, and how the amount of spatial overlap or agreement in these data sources might potentially affect whether integration will be useful. So for example, here if we have the primary data source 1 on the y-axis and some other data sources available on the x-axis, we might very well find that when we just plot out the data together, some might exhibit some negative relationship between the primary data source, some might have a positive relationship, and others have a neutral relationship. So there could be very different outcomes when we integrate these data. So I've largely put together what I'm calling this data check-in and ISDMs, and then this procedure for folks to be able to evaluate whether integration will be useful for them and their specific problem. I'm using mammals in North Carolina for this example because there are many, many data sources available just in North Carolina, or excuse me, the mesocarnivores in North Carolina. Still, state agencies such as the North Carolina Wildlife Resources Commission, they have a diverse set of information for many species, including hunter harvest records, deer hunter observation surveys, and trapper harvest records, along with other opportunistic observations of animals as well. And typically, most state agencies or management units are interested in monitoring the distribution or the abundance of some species, and they're just kind of left with the question of which data are useful and when should we integrate those data sources. So to visualize this, here again is the Candor Critters data set, just representing the presence or absence of bobcat across North Carolina. But we have, again, hunter harvest records, hunter observation records, trapper harvest records, and we have this for many, many species. And here, again, you can also probably see that there's some differences in the resolution of these data available. And as Krishna mentioned earlier on in the introduction, we kind of worked on developing methods to integrate data that have differing spatial resolutions in an integrated procedure. So quickly, I just want to illustrate and provide an overview of this three-step, what I'm calling a three-step data check-in procedure. And it starts with a data compatibility check. So here I'm suggesting we went through formal outlier analysis and evaluating a minimum sampling effort to just see whether the two given data sources are even compatible. And hopefully at this stage, we start to see some signals of whether two given data sources are going to be, are worthy or useful when bringing together in a single model. The next step of my data checking procedure is I go through a series of readily or very accessible preliminary data checks. So these are things like just correlation coefficients, linear relationships between data sources, and just evaluating the spatial agreement between these data sources. And these are also supposed to serve as checks for users to say, okay, I can easily calculate correlations between these data sources, and I find this given value, so this means, or this doesn't mean that I should integrate data. So the idea with the second stage was for it to be very easy and very accessible and give users an idea of whether integration will be useful for them. And then finally, in my three-step procedure, I go through a series of model fitting and evaluation where I fit all combinations of available data. I use a multi-objective optimization procedure to decide which data integration or which combination of data sources is potentially best for this scenario. And I evaluate the spatial agreement between single data source species distribution models and integrated species distribution models. And finally, we can kind of see, or results of this would look something like this. So in the top left of each of these species, we would have a single data source species distribution model, maybe just fit with the Candor Critters observations. But we see when we integrate in different data sources, our estimates of distribution are quite different. And this bottom figure is showing whether the estimates in the single data source agree with the estimates in the integrated species distribution model. So the white cells would represent the estimates agreed, and the brown cells would estimate or represent where they did not agree. So we see across different species, there's differences in the amount of spatial agreement between the data sources, and just pretty stark differences in the estimated distributions from a single data source versus these integrated distribution models. So this is a procedure that's very much in development, and we're excited to keep seeing this move forward. But just quick take homes for ISDMs, data checking in ISDMs is that so far we're finding it's surprising, maybe not so surprising, that it's going to depend on, unfortunately, the often unknown quality of the available data for whether they should be integrated. And so far it's appearing that the quantity of structured versus unstructured data definitely appears to matter, but it's been difficult so far to generalize across species. And of course, no single metric is available just to evaluate when you should or should not integrate data. It's going to take a series of metrics to evaluate this decision. And of course, ultimately, it's always going to depend on the objective of the study. And lastly, this bullet point just illustrates that there's usually always pressure right now with these new methods to always integrate more data. We have all available data, let's put it all in the same model and see what we can learn. But what we're finding in this procedure is that care should definitely be taken to ensure that it's actually necessary to integrate data, because you're going to get very different estimates when you do that, and maybe they're actually not going to be potentially representative of the species, how we might think. So I apologize for blazing through that last case study. I just wanted to quickly illustrate something that I've been working on in this dissertation. And again, I just want to thank you all for your time and patience in this talk and for attending this morning on Monday morning at 9 a.m. So with that, if we have time, I'm happy to, of course, answer